À l'unanimité également, le jury souligne les mérites de Madame Nassim Homayun-Sepehr et lui remet la médaille de l'Académie. Après avoir obtenu un diplôme de biologie à la VUB, elle a obtenu un mandat Télévie pendant 4 ans pour réaliser un doctorat sur la génétique de maladies rares du système lymphatique dans le laboratoire du professeur Vikkula à l'UCLouvain. Son travail est intitulé Identification of Genetic Causes and Molecular Mechanisms in Lymphatic Abnormalities. Ces travaux approfondis ont fait l'objet de 3 publications et il a défendu brillamment sa thèse en décembre 2021. Le jury ici voudrait particulièrement récompenser ce travail de recherche qui adresse la problématique de l'origine de maladies rares et complexes.
Comme vous avez déjà entendu, nous avons aussi Médaille. Alors, c'est le plaisir d'inviter Madame Nassim Homayun-Sepehr pour présenter ses travaux pour la médaille du prix ALVARENGA de Biologie.
Good morning, everyone. During my PhD, my goal was to identify genetic mutations and molecular mechanisms in lymphatic anomalies, such as cystic lymphatic malformations, Gram-Schlepp disease, and lymphatic-related fetal hydrops. Today, I will talk very briefly about each of them. Cystic lymphatic malformations can be like macrocystic, microcystic, or mixed cystic phenotype. They can appear as a pure lesion or combined lesions such as capillary lymphaticovenous malformation. Sometimes they can be part of overgrowth syndrome such as CLOVES. In this study, we analyzed for the presence of PRIX mutation in 123 patients, including common, combined, and syndromic forms of LMs. We found 180 PRIX mutations in 108 patients out of 123 patients, which accounts for 75% of our patients. 92 mutations were at hotspots positions. Here you can see hotspots mutations in dark colors. They are more frequent in common and combined LMs compared to syndromic forms of LMs. And interestingly, we found that variant allele frequency of mutations are higher in syndromic cases of lymphatic malformations compared to common and combined LMs. In this article, the general conclusion was that we found a statistically significant association between mutation types and phenotypes, and we indicate that PI3K pathway inhibitor drugs such as rapamycin could work in at least 75% of patients with a lymphatic malformation. I will talk about second disease, Gram-Schlepp disease. It's a complex lymphatic anomaly. Patients have retrograde lymph flow. They have osteolytic lesions, as you can see here, and sometimes can lead to entire bone loss. And here, if patients, the thoracic is involved, can lead to chylothorax, which can lead to death in some severe cases. There is no cure for Gram-Schlepp disease. At the beginning of my PhD, we had only one GSC patient. As I said, it's very rare disease. And we looked at variants in cancer genes. We found the KRAS mutation in this gene, and G12V is a very well-known oncogenic mutation. We confirmed our mutation by doing whole exome sequencing, and then we want to see what is the effect of this mutation in lymphatic endothelial cells of mice. Therefore, we reproduce control mice and mutant mice just to see the effect of this mutation. Here, these are the femurs of the mutant mice, and you can see lymphatics in bone. Normally, lymphatics in control mice, they don't have, normally mice, or let's say human, don't have lymphatics in bone. Therefore, we could see a feature of GSC in our mutant mice. During our experiment, we found that mutant mice die very fast, and it was because of chylothorax. Therefore, we looked at the lymphatic valves in our control and mutant ones, and we want to compare. Here you can see the lymphatic network in our mutant mice. They have fewer lymphatic valves compared to control mice. And as you can see here, the lymphatic valve is abnormal because normally lymphatic valves have V shape. And then we looked at the thoracic duct in our control and mutant mice, we injected a dye in our mouse, and we found that in our control mice, the dye was confined into the thoracic duct. However, in our mutant mice, the dye was refluxed from thoracic duct into lymphatics in the chest wall. We used a drug called trametinib just to see if it reduces GST features. And as you can see here, when we use trametinib compared to vehicle-treated mice, in our mutant mice, the number of lymphatic valves increased compared to our control mice. Therefore, we can use this trametinib, this drug, for Gorham cell disease, and also we just suggest that other RAS pathway inhibitors can be used for other complex lymphatic anomalies caused by mutation in the RAS-MAP pathway. And the last disease is lymphatic-related fetal hydrops, which is due to accumulation of fluid in more than 2 compartments, and it can lead to death. And 15% of these non-immune fetal hydrops have a lymphatic abnormality. 2 kinase inactive variants were identified in lymphatic-related fetal hydrops and central conducting lymphatic anomaly. In our lab, we identified loss-of-function mutations in this gene for CMAVM2. However, the molecular mechanism corresponding to each phenotype remain unclear. We looked at NGS sequencing data of over 700 patients with primary lymphedema. We found 30 novel variants in LRFH. We found 16 variants in CMAVM2, and we included references in our study because we want to compare with our new variants. This is the FB for protein, and we found, let's say, all of these mutations, which some of them are you see in red for CMAVM2, some for LRFH, and the rest, they act like a normal protein. We did a lot of analysis, Western blot analysis, FACS analysis, and we concluded our results in this figure. So normally, in a normal situation, FP4, which is a protein on the cell surface, binds to another protein on another cell surface, and it directs 2 different signaling, forward and reverse signaling. In LRFH variants, the proportion of this protein was higher on the cell surface, as you can see in blue, compared to normal protein, which we think that it might alter this signaling here, and it can contribute to in utero swelling of our fetus. However, in CMAVM2, the number of cells expressing the protein and the proportion of protein on the cell surface decreased compared to our wild-type protein. And we think that both reverse and forward signaling probably will be reduced, and maybe even loss of this signaling due to a second hit mutation. We need to further analyze what is happening in reverse and forward signaling for these 2 phenotypes. And I thank all people and all foundations for their support and help during my PhD. Thank you.